So you may have been left at the end of Friday's lecture wondering to yourself a question along the lines of why does all that meiosis crap matter? Because it's a lot to keep track of. It's a double division. You start with two of each chromosome, and each chromosome contains two chromatids in one cell. And then you end up with two cells, each of which has one of each chromosome with two chromatids. And you end up with four cells after a second division, each of which has one of each chromosome, each of which has one chromatid. Who cares? Why am I making you learn all this stuff? Think of this part of the course as putting jigsaw puzzle pieces together. Mendel had put some big ones together when he worked out that genes come in pairs. And of course, each gene can exist in multiple alleles. But you have two of each of your genes, and one of each pair is passed on to each one of your offspring. Now, Edward van Beneden has worked out this dance of the chromosomes in meiosis, and it ultimately is that chromosomes come in pairs, and one of each pair is passed on to each offspring in each egg cell and sperm cell. This is clearly important because when meiosis goes wrong, you end up producing an egg or a sperm cell that's either missing one of a pair of chromosomes or it's got an extra of one of its chromosome pairs. And the result is usually a defective embryo. Very often in humans, embryos with extra or missing chromosomes never develop at all. They're usually lost by miscarriage. In a few cases, you can be born with an extra chromosome, like an extra chromosome 21, but that causes Down syndrome. And you can occasionally be born with an extra chromosome 13 or chromosome 18, but usually people with that don't live very long after birth. So clearly this matters because if meiosis gets it wrong, the result is a fertilized egg that either never develops or doesn't develop right. And somehow the genes and the chromosomes seem to be following the same rules. In both cases, you get one of each pair. Everybody's got two of each, and one of each is passed on to each offspring. You got one of each from your mom, one of each from your dad, and you'll pass on one of each to any kids that you have. So what's the connection here? We know that genes aren't the same thing as chromosomes because you've got far more genes than chromosomes. There's something like 18,000 known human genes, and you've only got 23 chromosome pairs. So clearly a gene is not a chromosome. But in 1902, a couple of people that I'm not going to test you on, Sutton and Boveri, came up with the idea independently of each other that genes were physically carried on chromosomes, that a chromosome must be not a single gene, but a cluster of genes, much as pieces of music can be physically carried on lengths of magnetic tape or compact discs or USB drives or what have you. Genes are physically, whatever they are, their physical instantiation is on chromosomes. That caught the interest of a professor at Columbia University, still the only Nobel Prize winner to have ever graduated from the University of Kentucky, a fellow by the name of Thomas Hunt Morgan. He got interested in the problem. And Morgan and his students started studying genetics using these guys. This is Drosophila melanogaster, the common fruit fly. the thing that can take over your kitchen if you ever buy bananas and they get soft and you forget about them in your fruit basket. Never ask me how I know that. But you get these, all over. Uh, they feed on overripe fruits and vegetables. And Morgan and company studied them because, first of all, they're cheap. They're very simple to raise and they don't take up much space. Morgan and his students used to raise them in glass milk bottles. Milk used to be sold in glass bottles that you could return to the milkman when you drunk the milk. Sorry, just seeing chats coming in. It's just people putting in their names. He used to feed them on, overripe fruit that the fruit vendors were throwing away anyway. So you can raise Drosophila melanogaster by the millions in just one little room. They also breed very rapidly. They go from a laid egg to a sexually mature adult in 1 week. And the lifespan total is about 21 days. So you can have many generations of fruit fly in a reasonable period of time. And they also have a lot of traits that were shown to follow simple Mendelian rules. Differences in color, differences in eye shape and color, differences in wing shape, lots of traits that could be shown to follow the same basic rules that Mendel worked out for peas. It also comes in handy that Drosophila only has 4 pairs of chromosomes, and in certain cells, notably the salivary glands, for some reason, the chromosomes happen to be unusually large and thick. You can see the striping pattern on them. It looks like a very long barcode, and that makes them very convenient to study for various reasons. So there's some fruit fly chromosomes. You don't get this, but Drosophila does. They're called polytene chromosomes. So fruit flies are an example of something I've mentioned before. They're a model organism, which just means they're a species in which some important aspect of life that you'd like to study happens to be very easy and convenient. Elephants have traits that follow Mendelian rules, but it's simply not practical to do experiments on elephants because they take up an enormous amount of space, an enormous amount of food, and gestation period from fertilization to birth is, I believe, 18 months. They live as long as we do. So there's no real reason why you couldn't do Mendelian experiments on elephants. It would just be extremely expensive. You'd grow old waiting to get the results of your first cross, and they might also squash you. Fruit flies don't do any of those, and they continue to be used in genetics research all over the world. Sarah Palin was known to complain about why is the government spending money to study fruit flies. The reason is that there are an awful lot of things that we've learned and continue to learn about fruit flies that ultimately apply to us. One piece of evidence of all life sharing a common ancestor is the fact that organisms as different as fruit flies and humans still have a surprisingly large number of features in common that we presumably inherited from a very distant common ancestor back about, 2 billion years ago next Thursday. Right. 1909, one of Morgan's students, a guy named Calvin Bridges, discovered a mutant. Now, when I say mutants, you may well be thinking of the X-Men or possibly of, some horror movie monster like, the Swamp Thing or the creature from the Black Lagoon or what have you. To be more precise, a mutant is just one in which an allele changes from one generation to the next. It's what animal breeders used to call a sport of nature. It's just a change in an allele from one generation to the next that doesn't usually happen and doesn't follow Mendel's rules. We'll look later as to physically what a mutation really is. But for right now, let's just say that Calvin Bridges was looking at a milk bottle full of fruit flies, which are normally red-eyed, and he found one that had come out of seemingly nowhere that had white eyes. Well, fruit flies are fairly easy to manipulate with anesthetic and paintbrushes. So Bridges was able to isolate that white-eyed fruit fly and made it with a red-eyed fruit fly, and the offspring all have red eyes. So red eye color is dominant to white eye color. By the way, when we're talking about fruit flies, we use the concept of what's called wild type. Wild type just means what you would find in almost all fruit flies that you went out and collected in the wild. If you went to the back room of Harp's supermarket, where they store the fruit before setting it out, there's a pretty big population of fruit flies back there. Don't ask how I know. And if you collected a bunch of those fruit flies, you'd find that virtually all of them had red eyes. So red eyes are typical of what you find in wild fruit flies. And all of the phenotype that is just standard for wild fruit flies is called wild type. And wild type red eye color is dominant to white eye color. And so if you cross two hybrid flies from the F1 generation, you get 3 red-eyed flies for every 1 white-eyed fly. And this is exactly what Mendel's laws would predict. Looks like it's the same basic rules as for peas. There's a difference. With pea plants, they've got male and female sex organs together, so they don't have separate sexes. Most plants don't have separate sexes, although there are some exceptions. The one I can think of off the top of my head is kiwi fruit, has males and female plants. Fruit flies, of course, have separate males and females, and with a little practice, it's fairly easy to tell them apart. Differences in abdomen shape, and the male has some extra bristles on its legs. And it turns out that when you do that experiment and you sex the F1s, all of the white-eyed flies were males. None of the females were ever white-eyed. So if you do that cross that right there, what you end up with is 1/4 red-eyed males, 1/4 white-eyed males, and 1/2 red-eyed females, and never any white-eyed females. So there's something about white eyes that is much more strongly associated with one sex than the other. The only way you could get a white-eyed female would be if you crossed a white-eyed male with an F1 female, in which case 1/4 of your offspring are white-eyed females. And then if you cross that white-eyed female with a red-eyed male, you get red-eyed females and white-eyed males. Huh? What the heck is going on here? Why is there this odd association of a particular trait with one sex of fruit fly? Well, we said that in diploid cells, chromosomes come in pairs. That's almost true, but in many organisms, there's one pair of chromosomes that varies depending on the sex of the organism that has them. In fact, it's the sex chromosomes that determines the individual's sex. I'll make a little addition here. This can get more complicated, First of all, when I'm talking about sex, I'm basically talking about plumbing, about what glands and structures you've got for making babies. That's different from gender, which is a psychological and to some extent a social thing. So you can be born with one biological sex and yet psychologically know that you are of a different gender. This is just one of those things that happens. How it applies to fruit flies, I'm not sure. I don't really know what kind of gender identity fruit flies have. Given that their brains are so much smaller, I'm not sure it really applies. But in most mammals, and by an interesting coincidence in fruit flies, if you are of the female sex, you inherit two copies of a large chromosome that we call the X chromosome. If you're a biologically male human or a biologically male fruit fly, normally you inherit one X chromosome and one very small Y chromosome. So one of your pairs of chromosomes, if you're a male, is not really a pair. You've got a large X and a small Y. Those are some chromosomes from a female human. They took a female human cells, squashed them, stained them, looked for one that was dividing, and took pictures of the chromosomes and sorted them out like this. And of course, you've got the longest pair is chromosome 1, and the second longest is chromosome 2, all the way down to chromosome 22. And there at the lower right, you've got one pair of X chromosomes down there. You can see that they are fairly large. Those are chromosomes from a male human, prepared the same way. And you'll notice that 22 of those 23 pairs look the same between the female chromosome and the male chromosome. There's no difference worth talking about between them. But males have a single X chromosome right there and a single copy of a dinky little thing called the Y chromosome. We've not got time to get into what the Y chromosome does. Let me make it as quick as I can. Human embryos all start developing with a common set of plumbing. There's two pairs of tubes and a little lump of tissue that becomes a gonad, and various little flaps in the external genitalia region. If you're a female, biologically, one set of those tubes joins together and becomes your uterus and fallopian tubes. The gonad becomes the ovary. The other set of tubes mostly disappears, and you end up with female anatomy. If you are biologically male, the tubes that would become your uterus mostly disappear. You've got a tiny remnant of them buried in your prostate. The other set of tubes becomes spermatic ducts. The gonads don't stay in your abdomen, but they drop down and become your testes. And various little rearrangements in the between the legs department give you a penis and testicles and a scrotum, the bag that holds them. And you end up with typically male anatomy. The Y chromosome contains a sort of on switch that causes you to develop typical male anatomy. There's some fascinating things that can happen if something goes wrong with that switch, but you can end up with something called an intersex condition. We don't really have time to get into that. But the main thing that the Y chromosome does is switch on a pattern of development that gives you typical male anatomy. Morgan had gotten his idea to study fruit flies from a fellow researcher at what was then a women's college called Bryn Mawr in Pennsylvania. He had a job there before Columbia hired him. And one of his colleagues was the first uh, female scientist that I've had a chance to talk about. And it's kind of unfortunate that she died fairly young. She'd made the discovery that many insects also have chromosomal sex determination. Females have two X chromosomes, males have an X and a Y. That is not universal, by the way. Birds actually do it the other way around, where a male is XX and a female is XY, although never mind. A lot of reptiles, sex determination is environmental, where whether you're hatch out as a male turtle, for example, or a female turtle, has nothing to do with your chromosomes and everything to do with whether you were incubated at a slightly higher or slightly lower temperature. Quite a lot of fish are either both sexes, they're hermaphrodites, or they'll actually switch. So this means of determining sex is not universal in the animal kingdom, but it happens to be the one that most mammals use. And by a happy coincidence, it happens to be what fruit flies use. So by 1911, Morgan had hypothesized that the explanation for why you get those screwy results when you hybridize red-eyed and white-eyed fruit flies was that genes are physically located on specific chromosomes. Specifically, that gene for eye color was carried on the X chromosome, and that's a phenomenon that we call sex linkage. How does it work? Here's a Punnett square. The female up at the top has two X chromosomes, and so we can just write XX. The male has an X and a Y. So the female will pass on one or the other of her X chromosomes to her offspring, not normally both, although it can occasionally happen. The male will pass on either his X or his Y to each of his offspring. And because the X chromosome is carrying genes on it, we can write those as little subscripts. Now, it gets hard to tell a capital W from a lowercase w when you're using small superscript letters. So I've written the red-eyed allele as a plus sign. We often use plus to indicate wild type. And I'm using plus here so you can easily tell the difference. The male's X chromosome is carrying a white-eyed allele, and I've written that as a superscript W. So a red-eyed female has two copies of the X chromosome, each of which has the wild-type allele. She passes on one of those to each of her offspring. The male passes on either his white-eyed allele or his Y allele, and the Y does not have many genes on it at all. And the result is you get either red-eyed females that are heterozygous or red-eyed males, which have one copy of the red-eyed allele and one Y chromosome. But if you take one of those red-eyed females and mate it with a red-eyed male, you get this. A quarter of the offspring get X chromosomes with the wild type allele from both parents, and those are red-eyed females. A quarter of the offspring get an X with a wild type and an X with a recessive white-eyed allele. Those are red-eyed heterozygous females. A quarter of the offspring get an X with the red-eyed allele from mom and a Y from dad, and those are red-eyed males. And a quarter of the offspring get an X with the white-eyed allele from mom and a Y from dad, and those are white-eyed males. So you still get a 3-to-1 ratio, but this explains why you get that sex bias in the results. It's because the gene for eye color is physically carried on the X chromosome. It is what we call sex linked. By the way, in humans and in fruit flies, the Y chromosome is so small that it doesn't carry many genes on it except for that switch that starts you developing male anatomy instead of female anatomy, if it works as it's supposed to. There are lots of genes that everybody needs and that don't have anything to do with sex carrying on the X chromosome. That's why you can't have individuals that have two Y chromosomes and no X. That's why you have XX or XY, but you don't have YY. YY individuals would never be born. Um, in fact, there's no normal way you could get YY chromosomes because you could only do that if you had two males mating and each one contributes a Y sperm. You couldn't really do that without considerable amounts of genetic engineering. But there are no YY individuals. We all have at least one copy of the X. And there's lots of human examples of this. If you have normal color vision, you can probably see this diagram as the number 12. It's made up of orange-red circles and green circles, and the orange-red circles form the number 12 inside this field of green circles. But if you have the most common type of red-green color blindness, you probably can't see that this is supposed to be a 12. People with red-green color blindness can see other colors, but they can't distinguish red from green, which makes stoplights a bit interesting sometimes, to say nothing of picking out neckties. And roughly 7% of all men in the US have this, but only less than 0.5% of US women. I'm not going to ask anybody, but on several occasions when I've taught this class, at least one student has volunteered the information that he happens to be red-green colorblind. And every time I can think of, it's been a male. And usually there's at least one in a class like this. So it's quite possible that at least one of you is red-green colorblind. And there's that bias in sex because if you're a man, you only need one copy of the colorblind allele because your other chromosome is going to be a Y. So if you're a male, you inherit it from your, if you're a male and your parents are normal, you inherited it from your mother, and you only need one copy of the colorblind allele. If you're female, since you've got two copies of the X chromosome, and colorblindness is recessive, the only way for a woman to get it is to be the daughter of a colorblind father, because he's got an X with the colorblind allele, and of a mother that's either colorblind herself or a heterozygous carrier of it. Okay, there's fewer women than men that have it because you have to have two copies of the allele if you're a woman, but you only need one if you're a man. I have to bring in a cat example, of course. And you might have noticed I didn't mention orange cats. That's because the gene for orange color is sex-linked. It's dominant to the allele for no orange color. Orange is dominant to not orange. There's a neat little example of epistasis in action because if you have the gene for orange color, it blocks the action of the totally separate genes for black and brown color. So that's epistasis for you. But here's an orange cat. A male cat is either going to be orange or not orange. A male cat will either have an X chromosome with the orange allele and a Y, or an X chromosome that doesn't have the orange allele and a Y. And if he doesn't have the orange allele, he might look brown or black or gray or all of the things we've talked about. Females, however, can be heterozygous because they normally have two copies of the X chromosome. And if you have a heterozygous female cat, the cat will have patches of orange fur mixed with patches of fur that isn't orange, that might be black or brown or something else, depending on what other genes the cat has. That's a tortoiseshell cat. Here's a tortie right there. For your purposes, you can think of this as being like a form of incomplete dominance. This patched orange versus not orange coat. You can think of this as like incomplete dominance. And then if you have a cat that is heterozygous for orange fur and also has that gene for white spotting that we talked about on Friday, you have a calico cat. And there's a calico. Tortoiseshell and calico cats can only be female. The only exceptions are it is possible to inherit two X chromosomes from your mom and a Y from your dad. So you can have XXY and survive. In humans, that's called Klinefelter syndrome. One of the effects is that you're sterile. And this is true of cats as well. It's very rare, but you can get a male calico if the cat has two X's and a Y, but that cat is not going to have any offspring. That cat is going to be sterile. Barring unusual occurrences like that, torties and calicos can only be female, including the calico kitten that wandered into our house yesterday. For those of you keeping score at home, we've currently got 7 cats in the house. If anybody would like to make me a wholesale offer, I will be glad to entertain it. Right. Another famous human example, hemophilia is a disorder where blood can't clot or clots very slowly. People with hemophilia can lead normal lives, but they can bleed to death from even very small cuts. Have to be very careful if you grow up with hemophilia. There's several different ways you can get it, but the most common type, it's called factor IX hemophilia, is caused by a recessive allele that is sex-linked. So this is Queen Victoria of England. She was not herself hemophiliac, but we know she was a carrier of it. She was heterozygous for factor IX hemophilia. And I won't go into all of this, but here's her family tree. She was heterozygous, and one of her sons, Prince Leopold, was hemophiliac. He got her X chromosome with the hemophilia allele on it and a Y chromosome from his dad, Prince Albert, and he was a hemophiliac. Edward VII got her normal X chromosome plus a Y chromosome from Prince Albert. So he did not have hemophilia, neither did his son George V, neither did his son George VI, neither did his daughter Queen Elizabeth II. The British royal family today has a lot of problems, but hemophilia is not one of them. Leopold did inherit it, and he passed it on to his daughter, who passed it on to her son. Queen Victoria's daughter Alice got a normal X from Albert and a hemophilia carrying X from Queen Victoria. Just by luck of the draw, it's just, there's no rhyme or reason for it. It's just one of those things. Alice passed on her hemophiliac X chromosome to her daughter Alexandra, who married Tsar Nikolai II, the last emperor of Russia. And they had 4 daughters, and they didn't have hemophilia. I'm not sure if we know they were carriers or not, but their son, the crown prince, Tsarevich Alexei, was a hemophiliac, and he'd gotten it from his great grandmother, Queen Victoria. So he would have been Tsar, except the communists took over and shot them all. Um, he had severe medical problems caused by hemophilia, and his mother Alexandra sought out a Russian faith healer by the name of Grigory Rasputin. And you might have heard of Ra Ra Rasputin, lover of the Russian queen. Anyway, little history there. Queen Victoria's daughter Beatrice was a carrier of hemophilia, and she passed it on to the royal family of Spain, which had several hemophiliacs, including Alfonso and Gonzalo, uncles of King Juan Carlos. Alfonso and Gonzalo were not only hemophiliacs, they also liked racing fast cars, which is not a very good combination. So Prince Gonzalo right here died from internal bleeding after a car crash because he could not clot his own blood when necessary. So that's a famous case of a human sex-linked trait, factor 9 hemophilia. And yeah, there you've got Queen Victoria's daughter Alexandra, who's at the upper right, who married the seated gentleman Tsar Nikolai II and had 5 kids, Olga and Tatiana and Alexandra and Anastasia, the youngest girl. She's the one who's got her arms around the boy, Tsarevich Alexei, the crown prince, who would have become the next emperor if the Bolsheviks had not revolted in 1917 and had the entire royal family shot. By the way, I know you've seen the animated movie about Anastasia getting away, but it turns out they found the remains and she didn't. Yeah, hate to break that news to you, but Anastasia got shot in 1920 along with the rest of her family. And Alexei was a hemophiliac because Queen Victoria was his great grandmom. And just by dumb luck, he had inherited her X chromosome through all these generations, which happened to be carrying the hemophilia allele. Oofda. Okay, to make a long story short, too late, Wagner. Morgan and his grad students, Alf Sturtevant and Calvin Bridges, ended up winning a Nobel Prize for this work. They went on from there to demonstrate that genes are not only carried on the sex chromosomes, they're carried on the other chromosomes. And the word that we use for chromosomes that are not sex chromosomes is they are autosomes. So an autosome is just any chromosome that isn't a sex chromosome. Your chromosome pairs 1 through 22 are your autosomes, and Morgan and company were able to demonstrate that genes are carried on autosomes. And they used a neat little trick that I don't have time to get into exactly how they did it, but there's a way that you can very carefully do some controlled crossbreeds of fruit flies. Actually, it worked with other critters as well, but Sturtevant did it with fruit flies. And you can count how frequently chromosomes break and recombine during the crossing over process in meiosis 1. You might remember that I mentioned this, that one of the important differences between mitosis and meiosis is that in meiosis, in prophase 1, the chromosomes pair up. So each chromosome 1 pairs with the other chromosome 1, each chromosome 2 pairs with chromosome 2, and so on. And when they pair up, they do what we call crossing over. They break and rejoin. So each chromosome ends up with pieces of the other chromosome in its pair spliced in. So this is my attempt at making a diagram of how this happens. So for example, a gene for body color and a gene for eye color happen to be carried on the same chromosome. When those chromosomes pair up during meiosis, they break and exchange pieces. And now and again, that break happens between the location of the gene for body color and the gene for eye color. Sturtevant was able to measure how often that happens and was able to use that to map where genes are physically located on chromosomes. If we were in the usual classroom, I could point you to a wall chart that has thousands of human genes and where they are physically located on all of our chromosomes, all 22 plus X and Y's. Since we're not in our normal classroom, I'll just have to show you this. Sturtevant was able to use the frequency of that crossover to construct what are called linkage maps. This is a piece of a linkage map of the fruit fly X chromosome. Those numbers, they're a measurement unit called centimorgans. Don't worry about what that is. But they're calculated based on the frequency of crossover events. And they represent the location of each gene along the chromosome. So they come in an order. I used to compare this to the order of songs on a cassette tape, right? If you have a cassette, it's a long length of tape, magnetic tape, and there is a stretch of tape that corresponds to the first song on the tape, More Than a Feeling. And after that, there's a stretch of tape that corresponds to the second song on the tape, A Peace of Mind. And then after that, there's a third stretch of tape that corresponds to the third song. I forget what that is. It's been a long time since I listened to Boston's debut album. And so the songs are in an order. You know, there is first one, and then a second, and then a third, and then a fourth. I can't use that now because nobody uses cassette tapes anymore. In fact, I heard on the news that the guy who invented those died just recently. So I'm not sure how to teach this, but let's just say that genes have physical locations on a chromosome. There's a part of each chromosome that corresponds to each gene, and the genes are arranged in an order from one end to the next. And so here's the location of a whole bunch of fruit fly genes on the X chromosome with their relative locations along that X chromosome. So you can physically map where genes are located on chromosomes. That raises the question of what it is about the chromosomes that makes them able to carry genes. And we'll start looking at that on Wednesday with the work of a completely different scientist, a British bacteriologist named Frederick Griffith. But since the hour is getting late, I will stop the recording for now and we'll pick up with Dr. Griffith's work on Wednesday.